All right, welcome everybody. This is the Introduction to Biomedical Informatics workshop. This is brought to you by the Clinical and Translational Science Institute. I just want to go over a few things today before we begin. We have a whole library of workshops that we run throughout the year, both semesters, and they are available on our CTSI YouTube page for you to check out. They are regarding a wide variety of topics, as you can see here in this map. I'll paste the link in the chat. If anyone is interested in related topics, we probably have a video on it for you. For today's session, if you attend this session as well as 5 more in our summer boot camp, so I'll explain a little bit more about that in just a second, you can receive a certificate of completion and you can reach out to me, Elizabeth Bengert, ebengert@buffalo.edu if you're interested in earning that. So here's just the beginning of the list of the summer boot camp that is being offered. It starts in the beginning of July, runs through August. For the certificate only, you will have to attend at least 5 of these sessions as well as today's session. So our other speakers will talk more about this later. And then as well as continuing medical education credits, physicians are eligible to earn 1.5 credits for today's session. I'll be sending out a survey at the end of the sessions, so keep an eye out for that. And today's session is an introduction to biomedical informatics by Dr. Peter Elkin. But first, we're going to be starting off with some introductory remarks about the field by Dr. Ram Samidrala, and then followed by Dr. Barry Smith. So I will let you guys get started. Let me stop sharing. And go ahead and start us off, Dr. Samidrala.
Thank you. Thank you, Elizabeth. All right. So everyone can see my slides, right?
Yes.
So I'm Ram Samidrala. I'm a professor at the University at Buffalo and also chief of the Division of Bioinformatics. I work in the Department of Biomedical Informatics along with Peter Elkin, who's the chair of the department. The fundamental questions that my lab has been interested in asking and answering is, how does the genome of an organism interact with the information present in the environment to specify that organism's behavior and phenotype, and how can we use this information to improve human health and quality of life? And a lot of our early work was based on answering the first question. And then the second question is what will be the focus of what we are, this brief intro that I'm talking about today, and also to tee things off so that Barry can take it over, and then finally Peter gives a very nice introductory lecture on biomedical informatics that ties all of this together and the importance of it. I think that's a really great perspective to have. So I'm also part of I'm also the co-director of the informatics core at the Clinical and Translational Science Institute. This is funded by a very large grant called the Clinical and Translational Sciences Award, and this brings a lot of people together to make sure that clinical and translational science at UB is nurtured and encouraged. And then there's a lot of both research and both in terms of translational research, also translational science that's happening here. So in terms of our informatics core duties, this is just an overview of what we provide here for computational drug discovery. We provide informatics support for various kinds of researchers all over from the local area, from Buffalo to all over the world. And this includes proteomics, ontology, data analytics, and clinical and translational informatics. We have this multi-scale drug discovery platform that I'll talk about in a minute in a bit more detail. And then we are trying to develop actionable therapeutic candidates. We have some lead candidates for non-small cell lung cancer and opioid use disorder that we have filed. We've done a lot of in vitro and in vivo work, and we are trying to file some patents on it right now and trying to commercialize it. We've spun off some startups to try to commercialize it. We are developing this. The multi-scale drug discovery platform that I'll talk about requires a lot of high performance computing and programming and Unix knowledge to use. We are trying to come up with an AI-driven drug discovery interface that will let you use English terms and English language to query our platform and come back with answers. We also This is something I'm sure Barry will elaborate on, but we are trying to translate data for interoperability, combining, AI and machine learning and ontology standards and various interoperability frameworks to accelerate discovery. We have a variety of and the ontology part, I'll let Barry talk about later on in the second part. So for CANDO, this is a multi-scale drug discovery platform. Stands for Computational Analysis of Novel Drug Opportunities. And the basic idea is given in this figure. What we are trying to do is create these interaction signatures. So interaction signatures between what and what, you might ask. So between small molecules and every protein, every macromolecule in the system, that is every protein and every RNA. So we create a very large vector of values. Represents the signature or the behavior of these small molecules in relation to these larger macromolecules. And then we then scale that up, and then we say, what is the relationship of these small molecules and these macromolecules to the pathways, and then to at the level of cells, and then at the level of tissues and organs and phenotypes. We've implemented a lot of the signature work going up to the cell type, cell type, and then the cell, the overall organismal phenotype is also something that we are working on the other side using electronic health record data and so on. And so we are trying to integrate this very multi-scale drug discovery view in order to make drug discovery more efficient. Because if you look at everything else that's happening, it's related to technology. As computing speed doubles every approximately 2 years, then everything associated with computing technology tends to double also. But drug discovery hasn't capitalized on it. For example, next generation sequencing is becoming exponentially cheaper and exponentially faster. Whereas when you look at what drug discovery is doing, it's gone the other way, and so it's not able to capitalize on these efficiencies that we're getting in computing power. And so we want to make a difference there. And this is a more detailed view of how the drug discovery works. We start by generating these small molecule signatures. We do a variety of AI and machine learning and also some biophysics and biochemical algorithms to figure out what are the small molecules that best fit particular signatures that we desire. So if you have a particular set of interactions that we would like to have, then we can design a molecule, or we can find a molecule that will satisfy those constraints. And in terms of coming up with these signatures, we usually start from another small molecule. We usually have an existing drug, for example, that does something that we can characterize, and then we want to modify it. That's probably the easiest way, because we're dealing in a very, very large interaction space. And then the way we do the design is by deep learning. We have a couple of deep learning modules, and then that lets us engineer very novel molecules that are tailored in terms of behavior and function, rather than just in terms of structure, which is the more traditional view. And then finally, we go and validate all of this in high throughput discovery and measurement technologies that we're trying to come up with also at multiple scales. And then this allows us to calculate relate molecules to each other, hybridize them, combine them, and assess their effects on the phenotype or effects on the organism in a very multi-scale way. And so these are some results here. Like I said, for non-small cell lung cancer and for opioid use disorder, we've discovered very promising lead compounds that have been shown to work in vivo. So we synthesized 14 compounds for non-small cell lung cancer, 8 for opioid use disorder, and like I said, we always start with an existing compound to start with, because that's just because the nature of the signatures the signatures are very large. They're 20,000 vectors, or even larger than that, 20,000 real values. So these are results that kind of show that in the first one we are saying this opioid use disorder and binding to the various opioid receptors. And you can see we have a few nanomolar inhibitors here that are also antagonists, so that we are able to predict the function there. This is experimentally measured, by the way, but this is all based on predictions. And then on the right here, we have the rat experiments where we get rats addicted to fentanyl, and then we see how much of our see whether any of our drugs is able to reduce this fentanyl self-administration that the rats are trained to do in a given period of time or at a given dose. And we see at the maximum doses here for our top opioid use drug, then there's a reduction of fentanyl self-administration by 50%. And this is just the first try. This is just something that's computer spit out, and we synthesized it and we tested it. And we also show that in the last C, in the panel C and the very right, we show that this is not due to because the rat is become addicted to our drug. It goes back to normal habits. It goes back to reasonable food intake. So that's showing that it's the 50% at least is overcoming the fentanyl addiction. And this middle plot is non-small cell lung cancer. So this organization, it says this middle plot is small non-small cell lung cancer, and in the blue bars show you how much better our compound is relative to what we started with, the objective that we started with, and in control cell lines versus cancerous cell lines. And then the red bars show what is the relative superiority in the actual cancerous cell line relative to the control compound that we started with. And so in all these cases, these bars are going down. So this indicates the degree to which our compounds are doing better than the compounds that we started with. And there are 4 compounds being shown here. So there's 2 bars for each of those. So we are writing very detailed papers on this. So there's a lot of things, but this is just like a big picture view of what's going on. And finally, like I said, we are coming up with, so how can you use this platform? How can you benefit from using this? Right now, if a researcher in the CTSI comes to us to the informatics core and wants help, then we help them out by donating some of our time, or some of my students' time, and that's possible, but it's possible only for a low number of people. And that service is there, part of the CTSI website, so you can go find it on the web. But we are trying to come up with something called AI Can Do, where you can ask questions in English, like I've illustrated here, and then you can get back these kinds of answers. Or you can ask it, design me a compound for non-small cell lung cancer, and it'll do it for you. And then you can have to go synthesize it and go test it. So that's a work in progress at the moment. And I will, from there, I'll stop there, and I'll give the rest of the time to Barry.
Thank you, Ram. So I'm going to explain what ontology is. And this is a picture of the different databases which are spread across a very large organization. And the colors indicate the people within those organizations. The people with the same color are talking to each other, and they understand more or less each other's data. But as soon as you have people in different colored parts of the organization, they find it very difficult to share data. And this picture could be a picture of healthcare data organization modules within the CTSA members in Buffalo, or it could be a picture of the NIH, which is trying to deal with human health and disease, and which has lots of people collecting lots of data, but they're all doing it differently. And because they're all doing it differently, we can't combine the data. And that's what ontology is about. It's an attempt to solve this problem by creating stable, controlled vocabularies with stable logical definitions. So we want people to use the terms that they use and define those terms, and we'll help them to create the definitions, and then persuade other people who are using the same terms to use the same definitions. And this works. So it's been very successful, primarily in biomedicine at the turn of the millennium. It's now becoming more and more successful because of the growth of AI in a lot of areas. There's a kind of explosion of interest in ontology, and this is manifested in the activities of Palantir, for instance, which describes itself as the ontology company. So this works, but then it stops working. And the reason why it stops working is because when building ontologies to share data proves successful, other people want to have their own ontologies. And so we recreate the problem of silos which we had as between different modules within an organization. Now the problem of silos arises because different organizations build their own ontologies. And the reason for the success of ontology technology in biomedicine is because we built very early on this picture, which is a picture of how biomedical ontologies built for different purposes, different domains, would work together. So we have an ontology of organism types, which is a taxonomy. We have an ontology of cellular components. We have an ontology of phenotypic qualities. And each of these ontologies is built in the same way, using the same basic structures. And they all talk to each other so they can share the same definitions. And it took maybe 10 years to reach the point where we had a successful working foundry of ontologies. And this foundry of ontologies started off with maybe 20 participant members. Now it has something like 200 members. And the foundry methodology has been adopted in other areas, including by Palantir, which has what it calls an ontology foundry. All right, so this is a picture of the same structure, but with the structure made more articulate by calling it a hub and spokes structure. The hub is basic formal ontology, which is very general. It just contains of words like object and process, which everybody knows the meaning of. But then those words, object and process, get used by more specific ontologies. For instance, the ontology for biomedical investigations. And then we work further down to the spokes coming out of the spokes, coming out of the hub, which would be ontologies such as the yellow and orange and green ones at the bottom. And now there are, as I say, hundreds of ontologies. This chart is a very, very simplified version of the existing OBO foundry. OBO is Open Biomedical and Biological Ontologies, because all of these ontologies are completely open. Anyone can use them. All right, now BFO, which is the hub, is it's become an ISO standard, and it's used, indeed it's required to be used in over 800 organizations. So we are making progress. This is a Buffalo creation. We're making progress in solving the silo problem by persuading particularly large government agencies that they need to use BFO. And now it's been approved by several large government agencies, and it's now mandated for people who are building ontologies in those agencies, and therefore also for people who use the resources of those agencies and for contractors and so forth. And this is what I meant when I said that there was an explosion of ontology. All right, why is there this boom? Well, imagine that you want agentic AI, if that is still a thing. I don't know whether they decided not to continue using that particular buzzword. But anyway, if you wanted to do agentic AI across an organization like this, you're going to need a stable understanding of the terms which are used to describe the data in each of the modules in this organization. Otherwise, your agentic AI will be stuck in a silo, or it will mess things up by trying to cross silos without the benefits of having common terms with common definitions in controlled vocabularies called ontology. So that's why so many people now are becoming more and more interested in ontologies. Now, Buffalo is actually, by now probably along many dimensions, the world center of ontology, and it's pretty high up when it comes to biomedical ontology. And we have three great and prolific members of the biomedical ontology community in the Department of I have to get this right, the Department of Biomedical Informatics in UB. So this is Dr. Elkin, Dr. Coisters, and Dr. Deal. And now we also have a new biomedical ontologist, Richard Orbach, who he started calling himself a biomedical ontologist only in recent weeks, but he's been working with me on a rather ambitious ontology project connected with temporomandibular disorders. And we discovered that there are many, many people working on temporomandibular disorders across the world, and they all describe the various aspects of temporomandibular orders in the usual way, namely differently. There is very little in the way of consistent use and consistent definitions of the terms in this particular area of biomedical research. So we have grants which are supporting our work, but they're also supporting the work of several other hospitals and medical schools who are also working on TMD, and who are joined with us in making attempts to create a common way of understanding the data and using the data which is being collected across all of these different institutions. And now this brings us to the topic that I wanted to talk about today, which is common data elements. So when you're collecting data from patients, for instance, there are certain data elements of vital signs such as temperature and so forth, which you always collect for each patient. And it's good if these data items are collected in the same way, using the same terms in the different institutions which are doing research on the same disorder. And these terms are called common data elements, or to be more careful, a common data element is a question like, what is the value of, and then you would have something like patient's height or patient's pulse rate and so on, where the question goes hand in hand with the specification of how the measurement is taken and under what circumstances would one not take the measurement, maybe because the patient is giving signs of being overstressed and so forth. So these are common data elements. There are thousands and thousands of common data elements which are collected in the NIH Common Data Element Repository. And this is not rocket science. So these are some examples of common data elements. What is the disability status of this patient? What is the type of health insurance of this patient? These are the kinds of data that one needs to collect. And so the NIH CDE repository is a gigantic, and now I have to be very careful because this is being recorded. It's a gigantic, it's not a swamp, it's a mess. It doesn't work. And the reason it doesn't work is because we have, again, the silo problem. Every organization wants to have its common data elements accepted into the NIH repository. And so far, there have been very few controls which would be needed to ensure that there were consistent common data elements between different institutions. Now, the rules for building common data elements are not yet formalized in such a way as that the requirements established by the NIH in its data management and sharing policy can be satisfied. And this is proved by the fact that there are way too many common data elements which overlap in different ways, where what we would need is exactly the right number of common data elements so that we would be able to share common data elements across organizations, across disciplines, across disorders, and so forth. So if you go to the NIH repository and you enter the word injury, you will get 5,611 common data elements in return. So this is way too many. Injury is complicated, but the simple questions which need to be asked of patients which have specific injuries can be handled by far fewer than 5,611. The reason why we have 5,611 is that they overlap, but overlap in bad ways, which block data sharing and data exchange. Now, as it happens, there is a new scientific director of the NLM. It's the NLM which is responsible for the NIH CDE repository. That's the National Library of Medicine. And this gentleman is, apart from being a friend of mine, he is also one of the leading ontologists. So we have an ontology person now who is responsible for working out rules for common data elements, which can solve this problem of too many data elements. And he's working with UB ontologists and precisely with the TMD scientists I referred to earlier to create this set of rules for the creation of CDEs and for the use of CDEs, along with a benchmark set of CDEs for TMD research. And this will be the first set of CDEs built according to the new rules. Now, how can this work benefit the UB community? Well, we are in a good position because we can influence the way in which the CDE rules are both formulated and also put into place, because we are, along with Dr. Scheuermann in Washington, creating these rules, and we will be the first organization to test them. Now, that's our main focus of the other part of the informatics core, which Dr. Samudrala referred to earlier. There are other things that we're doing. We're building a translational science ontology, which is designed to formalize and standardize the ways in which translational science and the goals of the CTSA are captured when people are sharing data or talking about each other's work. Currently, we have a quite mature terminology, but we don't really have definitions which would ensure that the terms in that terminology are being used in the same way in different hubs of the CTSA world. And then we are creating a clinical and translational science ontology consortium, which is a consortium of CTSA hubs where there is an active ontology movement in the corresponding medical school. We have 10 hubs who are members, and the idea behind this consortium is that we will work to ensure that the same ontologies and the same CDEs and other features of data handling within the biomedical world will be shared initially across these 10 hubs, but then we will attempt to work out to other parts of the CTSA world in order to solve the massive problem of data silos. And I think that's the end, yes.
Thank you very much. If anyone has any questions, feel free to type them in the chat or speak them aloud. All right, well then in this case, I'll pass it over to Dr. Peter Elkin, who's the professor and chair of the Department of Biomedical Informatics.
Thank you very much. I appreciate the introduction, and thanks for those introductory comments, Dr. Samadrala and Dr. Smith. So I had told Elizabeth that I would start with the boot camp. So we have been doing a data science boot camp every summer for the last many years, really at the behest of our clinical informatics fellows. And this is the beginning of the training for all the clinical informatics fellows in the country, in addition to every single CTSA in the country, and also there are people at the VA who take it, and people from really around the world. Last year we had 743 people from 46 hubs, but 48 countries, 130 organizations in the U.S., and 191 government people took it. This year there are something like 33 lectures over July and August. We skipped the first week of August because it's epic training, and the fellows are usually there, so that can be a stop. I put up the QR code if you didn't get it from the chat and you wanted to get the link this way. That's a good way to get it. I'll leave that for a second. The topics are a range of topics, and you don't have to take everything. As Elizabeth said, that if you take 5 of the courses, and you don't have to take them synchronously, they're also recorded and put into a UB box, and so you're able to look at them at your leisure. Many of them have Jupyter notebooks, so you can actually put hands to keyboard and learn skills. And we would recommend that you, as you sign up for it at this location, then you can actually go ahead and get put onto the Jupyter Hub so you can do the exercises. No, we don't give you course credit for it, but the CTSA does give you a certificate if you do enough. I'm just going to scroll back to the end here, the beginning of the talk. So this is an introduction to the field, so it's kind of broad. We also kind of give you an introduction to UB and our department so that you guys are aware of it and can access it. So as basically was discussed, we have a division of clinical informatics, biomedical ontology, bioinformatics, public health informatics, and socio-technical and human-centered design. We have a lot of people that do a lot of work throughout the university, and we're involved in over 40% of the high-impact research at the Jacobs School. These are some of the bright smiling faces that you might see in the list of the people who are in faculty in the department, and we're getting more and more all the time. We have a bunch of postdoctoral fellows through. Some have gone on to become faculty or have left and gone elsewhere. We have a bunch of PhD students, and they also have, you can see some of their bright smiling faces here. And we have staff that are technical staff and administrative staff that are involved in this. And then we have a lot of volunteer faculty. There's a ton of them. And even people who've left the institution consider themselves still volunteer faculty in the department. So we have all kinds of interesting folks that you might find you may know by their just appearance. So our vision is a global impact through biomedical informatics innovation, and our mission is to continue to be a top 10 ranked biomedical informatics program in the nation, using our innovations to improve the global health of patients and their families. We are, I believe, in the medical school, the only top 10 department in the medical school. The other top 10 department in the university is philosophy, with Barry's work on ontology. Clinical informatics is a new medical specialty. It's approved by ABMS and is a subspecialty to any primary specialty. It's a 2-year fellowship. There was a grandfather pathway, but that's closed. People become CMIOs. And I'm just going to try to see if I can play this for you. I don't know if it's coming through loud enough.
Hello, my name is Dr. Peter Elkin. I'm the program director.
Oop. I paused that, and I shouldn't have. I was trying to make it louder.
For the Clinical Informatics Fellowship at the University of Buffalo. We have an outstanding range of cutting-edge research, very strong administrative experiences for our fellows, where they sit in the boardrooms of our hospital systems, along with the CEOs, CFOs, and CMIOs. We have a wonderful teaching experience with an outstanding faculty, and fellows are able to get their master's degree should they want to while they're doing their fellowship. We have a wonderful camaraderie between the fellows and the faculty, and a wonderful set of opportunities that will prepare you for every facet of clinical informatics practice.
Everybody in the program at the University of Buffalo Clinical Informatics is incredibly supportive. Any projects that you're interested in getting involved with, you'll meet with the program director, discuss your unique interests and goals, and then synthesize a plan over the course of 2 years as to how you can meet and achieve your objectives and goals. It prepares you very well to take the board exam when you're finished.
My interest in the program lies in the fact that we have such a wide range of areas of study, which includes public health informatics, pathology informatics, clinical informatics, and bioinformatics. In addition to that, we have a large group of outstanding informaticians who have an open door policy. They're always willing to help.
We rotated several of the local hospitals in one of the best regional health information exchanges in the country. So far as a first-year fellow, I've been able to work on projects that are analyzing COVID-19 patient data, reviewing physician alerts within the EMR. I'm currently working on a project with the Regional Health Information Change to look at population health data.
One of the projects that I was involved with at ECMC was a project that looked at data migration and data management. The hospital system was moving towards a new EHR in the ambulatory clinic setting, and this involved almost 20 different clinical sites. One of the issues that came up during one of the meetings was that of data migration. We weren't able to migrate all of the clinical data, so we had to figure out a way to define data migration on behalf of the hospital and all of the different specialties. I worked directly with the CMIO, worked with end users, attended executive leadership management meetings, and eventually figured out how, based on participatory design, we could develop a concise list of the data endpoints based on feedback from unique specialties. It was a great project to be involved with. It taught me a lot about project management, and it's something that I've developed skills that I'll take with me in my future in clinical informatics.
One of the projects that I am currently working on and I'm very excited about is looking at the impact of COVID-19 on telemedicine training in undergraduate medical education. Now, although telemedicine has been around for a few decades, with COVID-19 knocking at our front doors, we've all had to readjust. This includes medical schools having to readjust their curriculum to include telemedicine in training future doctors to use technology more effectively for improving patient outcomes.
Buffalo is a fantastic place to live. It feels like a big city, but everybody's very, very friendly. There's shopping, restaurants, nightlife, wonderful outdoor activities, hiking along the gorge, the Niagara River, and we're right next to Canada.
Now, if that sounds exciting to you, and you're a person who likes to take on projects, then the University of Buffalo Clinical Informatics program is probably for you, and we would love to see you here.
Should you have any questions, please do not hesitate to contact me at elkinp@buffalo.edu. We look forward to seeing you here in the spring.
Thanks, guys, for listening to that. I like that video. The fellows made it. They did a great job. So we teach in the undergraduate medical education, so the medical students, and there's a IMC 815, which is a clinical informatics rotation. We get about 2 students every block that come through that way, and they do very interesting projects. And then we also have research rotations that residents do with us. We offer a master's degree in biomedical informatics, a PhD degree in biomedical informatics, and including some MD PhDs that we've trained. We also have this ACGME accredited fellowship in clinical informatics, which you just heard a little bit about. And then we have postdoctoral fellowships in research through our NIH-sponsored T15 program. And also we have short-term training for biomedical informatics for undergraduates and master's students that's also funded by NIH. And we just started our undergraduate program in biomedical informatics. I think we're through our 5th class already. And we're now becoming a thread through the entire medical school education program, trying to work with all the program directors at each course to try to integrate 21st century medicine into the medical school education program. The T15 program has been very successful. In fact, we've had a number of supplements to it. We get 4 postdocs and 4 predocs every year, so they rotate quite a bit. So if you're interested in training in the science of informatics, let us know. And then we have a R25 program, which is for undergraduates and master's students. As I said, it pays $5,000 for 10 weeks in the summer. So whether you or you have a child that's that age, if they're from out of town, they can get a housing allowance. And this has to be for U.S. citizens and permanent residents, though that's all NIH will fund. And you also participate in the boot camp while you're there. This is a picture of one of our classes. They look very happy, don't they? Yes, thank you very much. And in fact, they are happy. We did surveys of them, and they seem to really be having a good time. We have a whole bunch of other awards. So we have active research in the department. The master's degree looks like this. I'm not going to go through it in detail with you, but just leave you with those slides so that you have it. The PhD program goes in each of the subspecialties that we have. And we have a lot of funding for the department that helps to fund all of the faculty and then the students and the research projects that we do. So this is my now Letterman's, for those of you who are old enough to remember, Letterman top 10 list of what biomedical informatics has done for you personally. So number 10 on the list is personalized medicine. So we worked with the pharmacists to come up with what the changes in medication should be when people have polymorphisms in their microsomal enzyme oxidase cytochromes. And it turns out this is the first intervention of personalized medicine that we've seen in the country, and this was actually implemented in RX norm, which is a drug terminology, and that's how it gets into EMRs, so those drug adjustments. We came up with e-prescribing. We worked with the pharmacists again to do NCPDP script, which also allows for drug-drug interaction and drug-herb interaction testing, and this has made prescribing safer. And there's an old joke about doctors' handwriting being so bad you couldn't read it, and now it's all coming in as digital, so the problem's eliminated. We implemented evidence-based medicine, so it turns out that you can't do evidence-based medicine without MESH, which is used to index the medical literature, and that's what's used in order to combine articles that are similar in their methodology so that you can do systematic reviews and, where applicable, meta-analyses to try to get summative evidence, and this led to clinical guidelines that leads to evidence-based medicine, and that's how we practice medicine today. Clinical decision support. So the error rate in medicine is unacceptably high. We need to do something about it, make it safer for patients and their families. And so we need to implement guardrails around the practice to try to make sure people are practicing best practice and not however they've been trained 20 years ago. And we want electronic health record systems to be better, and they're not great now. They're okay. We're implementing Epic. According to Bob Wachter, that's the cream of the crop. So there's problems still with the electronic health records, and they're getting better, and pretty much everything that's in them was originally developed in the laboratory and then went out to the industry. And I think that what I can see in the laboratory is that there are better things coming down the pike. Should be good. Translational research. So a friend of mine, Andy Ballas, in the year 2000, he wrote a book chapter that said that it takes 17 years to go from bench to bedside. That actually caught the interest of Congress and became a debate. They put now close to $50 billion a year into NIH. What are they getting for their money? And the next year they published an article with Elizabeth Boren, and this, by 2005, they had created the largest grant award that NIH ever gave, which is the CTSA award, which brings us here with you today. And then in 2009, that was so successful, they created the National Center for Advancing Translational Sciences, which is an institute at the NIH and center. It's a center at the NIH. And that is the people who give out the grants for the CTSA program now. And that all started because of a curious informatician named Andy Ballas. So we mapped the human genome. So in 2003, Clinton got up and said, we've mapped the human genome. Did we map everybody's genome? Nope. We mapped one guy's genome. He was an African-American fellow from Buffalo. That's whose genome we mapped, because Craig Ventnor was a professor at UB at the time when he took the samples. And this is the person whose genome was originally mapped in 2003 when they got it done. Now we've mapped more people, but we certainly haven't mapped everybody's. Then systems that facilitate paying for high-quality care, so pay for performance. So this is part of my work. We created methods to understand the quality of care that was being provided by a physician to their patients, and this was the basis that allowed the Affordable Care Act to put in the quality measures that led to the ability to do risk-adjusted quality scores for physicians. Those risk-adjusted quality scores became the basis for pay for performance, which allowed people to get paid better and also get paid for what they really do for complex patients, instead of sending people to take care of patients who are not sick. We created PubMed. So PubMed, literally, as it became open, Al Gore did not invent the internet, but he did make PubMed free for everybody to use, and that has changed access to the medical literature. So even within the time frame of a practice, doctors can look up evidence and use it to help their patients. But the most important thing that we're doing for you and your families is that we're systematizing the practice of care. We're turning it from a cottage industry where people practice the way they were trained into a systematized practice of care, and this is probably the most important thing that our field is doing for you personally. So how much should a clinician be expected to remember, and how much should the computer be able to provide decision support? Well, there are around 6,000 articles published every week, thousands of ongoing clinical trials, average clinical guidelines about 67 pages. The average new patient visit is between 30 and 45 minutes, and follow-ups are 15 to 30 minutes. Average elderly patient has 6 active clinical problems. It's 23 in my practice. And clinicians make hundreds of decisions a week. So what level of medical error and harm is acceptable to expect from a practicing clinician? Well, it turns out that that right now the level of medical error is about 6%, and about 17% of that comes to the patient, touches the patient, and we call that harm, and that's about 1% of doctors do. So 99% of what doctors do help you, but 1% hurts you, and I think we got to get that down. You know, if I told you that you're getting on a plane and you have a 1% chance of crashing, you'd get right back off that plane and you'd take some other mode of transportation. So what would it take to get to this parts per million or 6 sigma level of accuracy? Well, I mean, if you are on a plane, it's about 6 in a million is your chance of having problems. So we want to get medicine to that point. So we need to make use of all the tools available. We need to create clinical workflows that support best practice, not just minimal standards of care. We need decision support at the diagnostic level, at the workup strategy level, the treatment and the outcomes level. And then we need improved communication pathways, messages where you need them, when you need them, to the people you need them. So I'm on the NIH's AI task force and how to use it for research in the country. And one of the things that we did was we created a paper on how to use to try to identify and mitigate socio-demographic biases in machine learning algorithms, which are more and more taking over information provision for patients and their families, actually, as well as for doctors. We published this in a journal called Life, which is now a Q1 journal, and we found that there's much more than just algorithmic bias. There's all kinds of other biases that creep in, and we have to be on our guard to identify and eliminate them the best we can. This was published, first author was Gillian Franklin, an assistant professor in our department. Barry already explained ontology, so I don't have to do that to you. This is the ontology of general medical sciences, which is how they represent courses of illness. And underneath that, we have SNOMED CD, CT, LOINC, and RX norm. And then we build it into networks or sentences that actually expand it. we've taken these and built knowledge graphs, which are used in AI algorithms. And the idea was that a machine learning algorithm with an AI is superior to a machine learning algorithm without knowledge graph embeddings or general medical knowledge. So we went and we tested that in a database of 170,000 patients who had opioid use disorder and looked for overdose and death events. And we found that we were better able to predict it with the pre-trained embeddings. But much more interestingly, as we took data away from the algorithms, as we injected bad data into them, as we removed people from the algorithms, the top line, which is with the pre-trained embeddings, degraded much more slowly than the bottom line, which is without the pre-trained embeddings. So what that means is that by having general medical knowledge associated with our AIs, we can actually make them less biased. So we created the most accurate medical question answering AI in the world, better than Google, better than Anthropic, better than Microsoft. And the NIH actually said that right in front of Microsoft to them. They didn't look tremendously happy, but they took it. And basically the way we do this is you ask a question, it parses it into knowledge graphs, goes through our large language model, which expands it to be all the relevant associative knowledge. That goes into another large language model that actually provides the answer. We speak 243 different languages, and we actually have done this with a number of other models, and I'll tell you about that. She's kind of our mascot. When we asked an AI picture generator to generate semantic clinical artificial intelligence, they spit her out, and so she became our mascot. So I'm a clinician, so I like to start with a case. So I'll tell you about this case. This is a 24-year-old recent college graduate who comes to the office because of a 3-year history of increased frequency of urination and occasional incontinence whenever she travels. She says the urinary symptoms typically begin a few days before she's going to fly and stop the day after she arrives at her destination. She says she is anxious about flying. She reports having similar but milder symptoms prior to exams before graduating college, and her medical history was otherwise unremarkable. Her only medication is oral contraceptives. Vital signs are normal. Physical exam discloses no abnormalities. Urinalysis and urine culture are normal. So here are the possible choices that the USMLE questioner, the United States Medical Licensing Exam test, had as questions. And before our work, the LLM spit out, reassure the patient that the symptoms will resolve in time, which is the wrong answer. After we put in a lot of our knowledge base and ask the same question, it says recommend behavioral therapy, which is the right answer. And that just gives you an example of how we beat out the big companies. So this is the first paper we published in JAMA Network Open. It's a high-impact journal showing that we can improve at all sizes the original model's ability to answer medical questions. But what we found were a couple of interesting things. One is you got bigger and went from the 70 billion parameter to the 405 billion parameter model. It got better, but not statistically significantly so, which is interesting, actually. And we can see that we did pretty well in this. At this point, we had in the 70 billion parameter model, we still had a 8.9% error rate at that one. We have, at that point in time, we had 13 million facts in our model. We have increased it to 66 million facts in the model. And now our model got 98.4% correct, which is better than 99% of all doctors. This paper is under review currently. And here we tried the Gemini model, the Llama model, the Microsoft Copilot, and DeepSeek R1 distilled. You can see that we made them better, except for the Microsoft Pilot, who only allowed us to put in 8K of graph, and that wasn't enough to make a difference. But the other ones clearly got better. And interesting, and we like to point this out, that Gemini lost to Microsoft in their original model, but after we injected our knowledge graphs, they actually beat Microsoft handily. And I like to tell Microsoft this. They don't enjoy it, but it's a good point of information. So this is the QR code to get there, and this is what you'll see when you get there. And it automatically recognizes 243 languages. You can ask whatever questions you want. It doesn't. It's behind a secure server. It does not remember anything you put into it, nor does it remember the answer it gives you back, unless you click this little chat thing. Then it chats with you back and forth about it, but otherwise it doesn't. And so that can give you some, satisfaction that nobody's getting your data or your sensitive information. So I'm just leaving that for a second so you can put your camera up there if you want to grab the site and play with it. It doesn't cost you anything, and it's, like I said, used all over the world. So we get a lot of users. We did find qualitatively it gives more shorter answers than the Gemini model with Sky, so we're probably going to put that up too so you can use it, because that, I think, will be better for patients because it's more descriptive. But this was very good for clinicians. It gave you the right answer and just in a pithy sort of way. So this is the Gartner Hype Cycle. If you look at the Gartner Hype Cycle, we're up here right now. You can see my cursor. And the truth is, this is the valley of death. This is what happened in 1991, where AI went away. And what we need to do is get over here to stable systems. And the only way we're going to do that is with high-quality clinical trials. So we need randomized, prospective, double-blind, placebo-controlled trials that are able to help us to get from where we are today to where these things are part of clinical guidelines and part of evidence-based medicine practice. And so that's going to be the difference, and we need to do it quickly. So, I put in the question slide, but you can ask me questions at the end if you want. So I was going to talk to you about some of our other research, which we think is kind of cool. So we patented this. We think we can now make people younger. If you're a person that's like my age, you can see I'm not young. You might be thinking about the idea that you could be made younger. So what we did was we took a database that was available through the Broad, and we took people's in their 20s and compared them with 30s, 40s, 50s, 60s, and 70s. And what we found was that the Notch2 and Notch3 pathways were consistently downregulated over the years. And these are a pathway that is related to telomere length, which we know is related to aging. And then what we did was we traced that back to the root cause, and we found a molecule called furin, which had the same pattern. That was the first thing in the cascade. And then we also found some things that built up. As we lost furin, we lost GZMB and HES1, and that caused them to go up. And when they went up, those built up toxic products in the cell and may lead to dysfunction. So we saw that in younger cells, they made good furin, but in older cells, they didn't. And this is statistically significantly different. And same thing with Notch2 and Notch3. So then what we did was we transfected the older cells with furin, and we were able to make muscle again. So here's the young cells, older cells. Here's the, oh, I've got to put my cursor over here, I'm sorry. Here's the young cells, they're making muscle, older cells, not so much. And then after we transfected it with the gene product for furin, we got muscle again. So we actually turned back the clock of time for them. And it actually happened in 5 days. So we were able to do this and get statistically significant results. So if you look at it, at 3 days we start to get an effect, but in 5 days it was as good as when you were young. I think it's pretty cool, actually. So we patented it, and now we built the drug, and now we're testing the drug itself on it. And then after that, we're going to go straight to clinical trials and get breakthrough drug authorization by the FDA, and also we'll also try to test it for Alzheimer's disease, and I'll tell you a little bit about that in a minute. So if we look at this is GZMB in young cells, older cells. After we transfected it, came back down. This is a bad breakdown product that builds up in the cells that we want to get rid of, that's causing dysfunction. And this is also true in Alzheimer's disease. So as furin goes down, ADAM10 and TACE go down, and beta amyloid go up. And beta amyloid is what builds up in Alzheimer's disease. So we think we can maybe do something in neurodegenerative diseases with this as well, and we're going to test it as a breakthrough drug, we hope. And I already have an appointment at the FDA, so that ought to make it a little easier. So we did work on the cure for cancer. We don't want to do little things anymore, so I'm going to tell you about how to win the race against cancer. We think we may know how to beat cancer in general. And so we published a couple of papers on metformin, and metformin actually had a 18% 5-year survival advantage in non-small cell lung cancer. This is a drug you take for diabetes, and what it does is it takes sugar out of the bloodstream and pushes it into muscle. So we thought that this might be an opportunity. This is a graphic from one of our papers. And we thought that this could maybe be a useful thing in cancer. So what we did was we took cancers, and we had both squamous and adenocarcinomas, and we grew them in increasingly smaller amounts of sugar. And as we did that, you can see that they grew more slowly, right? And then what we did was we added back ketones, which is what, when you go on a low-carb diet, that's what your body makes. And your body can burn ketones. It can learn to burn ketones. But it turns out the cancers, except for the most early stage cancers that were adenos, not the squamous ones, they were able to do a little bit, but not much. And then we looked into a little bit about why that was so. And it turns out that AT1 and OXCT1 were down, and those are the things that are necessary in the ketolytic pathway, so they can't burn ketones. This is a fundamental difference metabolically. And I was talking with the National Cancer Institute just 2 weeks ago, and they're very excited about it. They think that we may have something that could be a big differentiator. They also are interested in Roswell in the muscle thing, because a lot of people discontinue their chemotherapy because of sarcopenia or loss of muscle. So if I can give muscle back to those people, they may be able to complete their treatment and cause them to be able to have greater survival, especially. So we're looking at working with them to do those experiments as well. So I'm editor-in-chief of a couple of journals, just so you know. The Journal of Translational Research, since we're in CTSA, it does translational research. That's my journal. It's a Taylor and Francis journal, and it has a worldwide reach. Please send your best work to us. And I'm also editor-in-chief of the Journal of Biomedical and Health Informatics from IEEE. That actually is the highest impact journal in the field. It has an impact factor of 7.7 now. The new impact factors just came out. And so if you have really great informatics research, please do send them here. We would love to see it. Whoops, sorry. Here's me going too fast. I'm also part of, I'm the co-chair of the Best Practices Group for the Learning Health Systems CTSA Partnership. So if you're interested in learning health systems and CTSAs, please get in touch with us. We'll get you involved. There's a little QR code if you want to learn more about it. And this is a very important thing for the CTSA consortium in general. I'm also on the board of ACTS, that's where I was before this, which is the Association of Clinical and Translational Sciences, and we're the academic organization of the CTSAs. And so there's an awful lot going on, and please do get involved. If you like ontology, Barry and I and others wrote a book that's apparently a bestseller for Springer Nature, terminologies, ontologies, and their implementations. And I think in the next one it's, maybe that's still what it's called. No, second edition, yeah. So that's what it's called. Please consider reading that book if you're interested. I don't get any royalties from it. They paid us a fixed fee at the beginning, but it's really a well-done book, and it came out much better than we thought, and we had wonderful people involved in creating it, so including Barry, if he's still here. Yep, he's still here. I did that already. Then this is just a picture of one of the people who came. You can see Dr. Samadrala is there, and his mentor, Mike Levitt. And I'll just close with a couple of quotes and take any questions you might have. Machiavelli in 1505 said, "There's nothing more difficult to take in hand, or more perilous to conduct, or more uncertain in success than to take the lead in the introduction of a new order of things, because the innovator has for enemies all those who've done well under the old conditions, and only lukewarm defenders in those who may do well under the new." And this is just as true today as it was in 1505. So, 500 years later, and we're still in the same boat. And then my favorite quote's from Peter Drucker. He says, "The best way to predict the future is to create it. Come create the future with us." This is Mont Saint-Michel. It took 1,000 years to build, and the third and fourth and fifth generations that worked on it knew for sure they'd never see the full result of their labors, but they did it anyway. So my challenge to you is, don't pick low-hanging fruit. Do research that's big, hairy, audacious goals. And even if you don't solve the whole thing, you'll make a dent in it, and people will build on your work, and in the end, you'll be part of something beautiful like this monastery on a mountain that we get to see these days. So with that, I will hit escape and stop sharing, and be happy to take any questions that anybody might have. Thank you all so much for coming and for taking the time to listen to me.